Welcome to this latest edition of our online discussion series for the project Pushing the Boundaries, which is co-led by me and Tarja Knutila at the University of Vienna. And we're interested in organisms, agency, evolution, AI. For this series, this is kind of an outreach activity of the project. We invite people that we find interesting, that we are reading, whose work we are using, and we ask them to present a quick introduction to some question or some topic that they want to discuss. We have a panel here that will then continue the discussion for the next 40 minutes or so, and on the full hour, we'll open the discussion to everyone. Welcome, first of all, to Jannie. I'll introduce Jannie in a second. Welcome to everybody else. If you have questions, you can either put them in the chat, or afterwards, when we open the question to the audience, you can switch your camera on. We are recording this, and we will put the discussion on YouTube. So if you don't want to be seen, just write in the chat, and we'll read the question for you. A few quick things that we always say, but shouldn't be necessary. Do raise your hand for a question so we can see you. If you want to ask it yourself, of course, stay civil. Go for the argument, not the person, and stay on topic of the discussion, please, if that's possible. It's going to be a challenge today, but we will try to avoid jargon and technical concepts, but some of them will sneak in, because this is, after all, a discussion about modeling. And that brings me to our guest today. And it is a special pleasure to have Jannie here, because Jannie, you may not know this, is the reason I am studying agency and the organization of organisms today. We spent a year together, a wonderful year in Berlin at the Institute for Advanced Study. And it was Jannie who got me, I was interested in this topic before, but who got me really going during this year on this topic. So thanks for that. But actually, Jannie is famous for doing something really funny, for taking an economics theory and applying it to biology, to biochemistry, which is, I think, a unique thing. And the idea is that metabolism is usefully modeled by supply and demand formalisms from economics, which was kind of revolutionary and a much better approach than the sort of steady state chemical network modeling approach that other people had. That is one of Jannie's claims to fame. The other one, which we will talk about today, is that he took the beautiful work of Robert Rosen, which is very hard to understand and was never connected to biological reality, and extended both Rosen's formalism and also adapted Rosen's model to an actual biochemical cell, because, and I should have mentioned that before, Jannie is, of course, a biochemist and a very good expert on all that. So we'll talk about this model, this relational model of the cell today, what it means to capture the behavior of a whole living cell, and how Rosen's concepts map onto the actual biochemistry of a living organism. And without further ado, I will just hand over to Jannie now and his introduction. Welcome, and it's all yours. The floor is yours.
Thanks, Yogi, for that introduction, and welcome everybody else. It's wonderful to have such an audience of experts and people that are interested in these topics. As Yogi said, we share a long-standing interest in all of this stuff. So let me begin by asking a question. And it's a very interesting one. I mean, we are all, as living organisms, made of stuff that is fragile. So in a year's time, most of the atoms that you consist of will be gone, and they will be replaced by others. Yet you, as a system, as an organism, as a self, still persists. One can put it, the question is, how is it possible for a system to live longer than the lifetimes of its components? So how can you be persistent when you are fragile? So that just as an introductory question, because that's the one that I actually want to answer. But to answer that, we have to really go into what happens inside organisms. And I'm going to focus on a single cell. I don't want to complicate things too much. So think about a yeast cell or a bacterium, sort of single living cell that I will use as my model organism. So I'm going to share my screen. I'm afraid I can't do without pictures. I'm a very visual person, and I think you'll see that it's impossible to explain these things just in words. So bear with me. It's going to be a little bit of a lecture, but let's see how it works. So, Yogi, can you see that? I've shared the screen.
Perfect. Perfect.
Okay. So, this is any biochemistry student's nightmare. This is the so-called metabolic chart. Supposedly all the chemical reactions of metabolism. And I want to focus, I want to zoom into this to try and find out what the core of this metabolism is that will allow me to start answering the question of how is a cell able to make itself, because that is, of course, how you are able to live longer than the lifetimes of your components. You have to autonomously make all your components. So let us do a serious zooming in. This is the core reaction network of metabolism. So what all of us, all organisms, take in nutrients, and intermediary metabolism basically breaks down these nutrients into three groups of, they call them building blocks for macromolecules, for the big molecules. So there's amino acids on the one hand, nucleotides, and lipids. So amino acids are used for the production of proteins through polypeptides. Nucleotides are for the production of all the different types of polynucleotides in the cell, DNA and transfer RNA, messenger RNA, ribosomal RNA, and then the host of what we now know, non-coding RNAs, which play a regulatory function. The amino acids, through their coupling with tRNA in the genetic code, they are used for the production of polypeptides, and this is going to be the main focus of what I start doing. I'll later on bring in what happens from this side, but you can see polypeptides use the information from messenger RNA, the sequence information of nucleotides, and translates that into polypeptides. So this can be regarded as a process. We can actually depict the process as I did up here, nutrients in just purely as a process that produces polypeptides. I'm going right from there to the polypeptides. But there's a problem here, is that we don't know what makes this happen. And for that, I have to first make a little detour. And the detour is to a genealogy of what's called relational biology, starting with Nicholas Rashevsky, who had as a PhD student Robert Rosen, who has as a PhD student Aloysius Louis. And the three of them, through a matter of decades, have produced now a corpus of knowledge called the relational biology. And I need that in order now to start making sense of what happens inside the cell. So let's just make a quick detour. In relational biology, everything is modeled as a set. Every process is modeled as a mapping. So a mapping is something that maps the elements of one set to the elements of another set. And these are two ways of depicting that. However, there's a much more useful way of doing this in terms of what's called a graph theoretical relational diagram of such a mapping. So this is the map F that maps set A into set B, or if you want to only look at one of the elements of A and how it maps into one of the elements of B, you can also use this diagram. In this form, it's called an element tracing diagram. So this is the sort of formalism that is used in relational biology. It is the basis of what is called category theory. You can do lots of things with these mappings, and as I'll show some of it. But what is important and why I went from this a lot to the relational biology is that Rosen realized that you can actually use the Aristotelian causes, or as I prefer to call them big causes, they are actually more explanatory factors. And you can use these to understand a little bit what's happening in such a mapping. So what he said is that basically the efficient cause is what makes things go. So by what or how things happen, this is from what something happens, and this is what happens, the outcome of the effect. You can think of this as a processor that acts on an input to give you an output. But what he said is that you could use these, Aristotelian causes to map onto such a mapping. So, of course, there's also another way of, one of the causes is the final cause. And if you look at this, you realize that the function of this F is to produce B from A. And on the other hand, the purpose of A is to be that from which B is produced by F. So if you think of these as the means of the process and this as the end of the process, a sort of a theological way of looking at it, then the effect is actually the final cause of these, of the means, so of the efficient and the material cause. So B is therefore the final cause of F and A. But this was very important because this allowed you to start dissecting the types of big causes or explanatory factors that you need for explaining what happens inside the cell. So what I left out from my previous view is actually what is the mapping in full? What are the processes that make this process happen? This conversion of nutrients into the various polypeptides and so on. These are all in the cell are enzymes. So they are catalysts that catalyze these reactions. That's very important because if you again think of that metabolic map, there are thousands of other, millions of other reactions that could possibly happen, and something has to pull the metabolic map onto a faster time scale so that it's kinetically isolated, so that you only have those reactions happening. Now, most of these biochemical reactions don't happen at all or very slowly, so you need catalysts to increase the rate at which these reactions happen. And these are then, for these particular processes that are depicted here, are the enzymes. And then ribosomes are the, let's call them molecular machines, I'll come back to that later, that actually synthesize the polypeptides. So there is a first, what I would call an efficient cause inside the cell, or a set of efficient causes inside the cell that can be seen as a group. All these reactions that happen inside the cell are changes in the type of bond between atoms and molecules. They are so-called covalent bonds, strong bonds where atoms share electrons, in contrast to weak reactions that we'll see, weak interactions that we'll see later, so-called non-covalent interactions. So this is why I call it covalent catalysis. So that is the first set of efficient causes inside the cell. What happens to these polypeptides? Now, remember, a polypeptide is a string of amino acids. So all the important there is the sequence of the amino acid. It has no function yet, the polypeptide. In order to be a functional protein, such as these enzymes, what has to happen? It has to fold into a three-dimensional structure, or you can even have more polypeptides self-assembling into higher order structures. To go from there to there is to get an activity. I call it the functional entailment. So you go from a non-functional molecule, the polypeptide, the long string of amino acids, into a three-dimensional folded active functional structure, such as an enzyme. And that is extremely important. And so what causes that? And this is something that biochemists usually don't regard in their models, but I'll show that it's absolutely crucial. In order for this folding to take place, it can only happen within an environment that has very specific properties. It has to have the right pH. It must be watery in the first place. It has to have the right pH. It has to have the right ionic composition. It has to have the right ionic strength. Usually the fact that there's a high protein concentration also has an effect on the environment. I call this the intracellular milieu. And it has to be absolutely right in order for those polypeptides to fold into a functional structure. If you change the intracellular milieu, for instance, you change the pH, or you start adding something like ethanol, or you do it in chloroform, these polypeptides, they may fold or they may not fold, but they won't fold into a functional structure. So in fact, the intracellular milieu is like a catalyst. Because if you change it, it speeds up the reaction, and if you change it, it will have an effect on the functionality of the proteins. So I think this is something that is new in my model. Nobody else has really regarded the intracellular milieu as a efficient cause. So these processes, these folding processes, and the stability of the proteins are held in place through weak interactions, which I just called non-covalent interactions. The process is now called supramolecular chemistry. This was Jean-Marie Lehn in the 90s, got a Nobel Prize for this. And this is very important. In fact, if I were to ask you what is the secret of life, well, the secret of life is that it learned to harness these supramolecular processes. Right, so we have a second set of efficient causes. So the first set made the polypeptides. The second efficient cause actually then functionalizes the polypeptides. And now the question, of course, remains, what keeps the the intracellular milieu at the right composition? And for that, one looks at transporters that are in the membrane of the cell that selectively transfer nutrients on the one hand, but especially electrolytes, ions. And if you look at the composition, for instance, of the inside of the cell and you compare it with seawater, you see there's a huge difference, especially in potassium. If you look at the difference between there and there, and in sodium, low in the cell, very high outside, chloride, low in the cell, high outside, and so on. And then amino acids and proteins, very high, of course, nothing outside. Calcium, very important, low calcium, high calcium in seawater. So in order to keep the intracellular milieu at this very specific ionic concentration, you need electrolyte transporters that sit in the membrane that then transport in and out the electrolytes. Now, these electrolyte transporters are also polypeptides or proteins that are made from polypeptides, so we have already accounted for them. So you can see I've got a mapping up here that these are the electrolytes, the electrolyte transporters that then transport the electrolytes into the intracellular milieu. So what we have are three sets of efficient causes. And what you can see is we can glue them together into a particular structure that is closed to efficient causation. That is Robert Rosen's seminal idea. All the efficient causes, the C and the T and the M here, the catalysts, the transporters, and the intracellular milieu are all created inside the system. There's nothing from the outside. It's autonomous with respect to its production of itself. So this is a very important little model that comes from my analysis of what happens inside the cell, the biochemistry of the cell. So I started with the biochemistry, and then I created the model. This is important. I went from biochemistry to the model. So if I put them all together, all of these things together, I have a view of what happens in the cell, a sort of a bird's eye view that focuses only on the important interactions. Of course, there's thousands of other things that happen inside the cell. There's a huge regulatory superstructure sitting on this to see that all the kinetics of the processes are also fine-tuned to each other. So there's a lot going on, but by focusing in on this, we get to the core of what is needed in order to be close to efficient causation. So this is a view, biochemical view of a cell in terms of its efficient causes and how they are dependent on each other. So let's get back to Robert Rosen. So what he did is he did magic with these mappings. Of course, what can happen is you can compose these mappings. Also, of course, in category theory, you can compose such mappings. You can form cycles of these mappings. So, for instance, here you have a cycle that all the efficient causes sit on the outside, all the efficient causes here sit on the outside. So this one could call material entailment. And now, important, if one reads Rosen, he calls such a thing a machine. Well, this would be a composition of two machines, or this would be a cycle of three machines working together. It's a very specific way of using the word machine. Of course, it's used in different ways in other disciplines. But what is important is that the mapping itself, the process of this mapping, can actually be in what is called the target or the codomain of such a mapping. So you have a hierarchy of mappings where a higher order mapping produces a lower order mapping. So in fact, you can call this functional entailment. This particular mapping functionally entails this mapping in order to do this at the bottom. This he calls a mechanism. So anything, if you think one way of looking at it, let's say this is hardware and this is software, and this is both hardware and software. In this mapping, it's software, in this mapping, it's hardware. So he uses that idea to delineate this type of mapping from what we have at the top here. So this is what he calls a mechanism. And what can happen in such a mechanism, if you identify these two, G and B, you can form a cycle which is called a hierarchical cycle, which can you see is close to efficient causation. All the efficient causes are produced inside this cycle. And this is what he calls a complex system. So any system that has such a type of hierarchical cycle, which is close to efficient causation inside it, embedded inside its network, he calls a complex system. And if all the efficient causes are inside such a cycle, he calls that an organism. And we've already now seen one example of an organism, the cell, that is close to efficient causation. And this allows me now to get to this important point. So he had this idea of functional entailment, but what he saw is that if you identify this set with that set, you get such a structure, which he said is a very good model of what he saw happening inside the cell. He said, look, there's stuff that's happening in the cytosol of the cell, all the metabolic reactions, which he can model with this particular mapping. However, we know now that the enzymes are proteins. They have a limited lifetime. They get dysfunctional, they get broken down, they get taken out of the cell, so they have to be replaced. And he said, well, there's a possibility of using the products of metabolism in order to manufacture what is missing here, which has gone out of the cell or become dysfunctional. He called that repair. Perhaps better to call it replacement. And the Santiago group of Letellier and Cornish-Bowden, they also actually rather use replacement instead of repair. However, this is not close to efficient causation. You can see there is still one efficient cause that sits outside the system. And this is where now the crux comes. In order to repair the repairer, you need another mapping. And so you can add such a mapping, in this case, the mapping H, which then uses some other material D in order to make that mapping. But of course, now you have another mapping sitting outside the cell. So if you want to repair that one, you have to add another mapping. You get an infinite regress. So Rosen asked himself, is it possible to pull this mapping H inside into a system so that you have a system that becomes close to efficient causation? And he found a way of doing this, mathematically at least. And people spent lots of articles, paper on arguing about this particular system and saying that it's wrong, but it's not. Perfect. The mathematics underlying this incorporation of the mapping H into this, what he calls a replicative MR system, is perfectly sound. However, he was never able to map this model onto reality, onto what happens inside the cell. Nobody else has been able to do it. And this is always a problem. It's called the realization problem. He wasn't able to realize this model in terms of reality, in terms of what happens inside living organisms. And for me, this has been a problem. So the snag, of course, is that you have the products of metabolism being used to repair, to be the material that repairs the mappings. But at the same time, they must also be processes themselves. And so this dual causality, dual causal status is a huge problem inside this model. You'll recognize this as what I've already seen comes naturally from looking at what happens inside the cell. So this is another type of model, which is also close to efficient causation, and which I will now show is actually the model that is useful for describing self-manufacture of living organisms, of cells. So this I call the fabrication assembly system. But immediately I must show that there's a problem. And this is why modeling is so useful, because the formalism forces you to think about it very carefully. So you can see here's a problem. F has to do two things. It has to map A into B, and it also has to map F into D. So in fact, this is not possible. A mapping can only do one thing. So I have to split that into two mappings, the sum of two mappings. So F2 maps D into G, F1 maps A into B. But this causes a formal problem. G cannot map B into two things. A mapping only maps into one particular set. So G will also either have to be G1 plus G2, or I have to split this. The material cause for these mappings, I have to split into two, B1 and B2. Then G will map B2 into F2, and G will map B1 into F1. And it seems that, yeah, this is nice. We can now separate these two. This is a mechanism, can you see? It's the same as that functional entailment, metabolism, repair mechanism. And this is a hierarchical cycle. But if you, again, look at it very carefully from a formal point of view, there's a problem. F has to map A into B1, but it must also map A into B2. So again, you have this problem of one particular mapping having to do two things, mapping A into B1 and A into B2, and that can't be. So in order to get out of this, I'll have to split F1 into F11 and F12. And this leads to this, again, an infinite regress. I have to keep splitting. And the question is how to solve this infinite regress in the model. And this is, again, I want to emphasize, it's by looking formally at the model that you suddenly start realizing this thing. And this brings us to the elephant in the room. Up to now, you'll see I've only used 3 of Aristotle's 4 causes. And in fact, relational biology, Robert Rosen and everybody else who's argued endlessly about Rosen's models have never really used formal cause. Rosen mentioned it here and there, but he didn't use it in his models. And I'll show that it is absolutely essential in solving this infinite regress. In fact, it's essential in models. We can't do without the idea of formal cause. How are we for time, Yogi?
We are hopefully nearing the end, but you're doing a really good job.
Okay,
yeah, with a lot of information in very little time. So go on.
Yeah, so,
don't worry about it.
Let's soldier on. Okay, so what is important is that up to now we've been talking about B as the effect of the mapping, but in real terms, B is a substance, also in Aristotle's idea, in Aristotle's cosmogony. So a substance is always a composite of matter and form, and we've only been looking at the matter of the material. So this is Aristotle's hylomorphism. And so what process F actually does, it not only enmatters it matters B from A. This is a term that I think is very useful to learn. It's also, it informs. It provides the form of B. So enmattering is actualizing a substance in matter. So the efficient cause uses the material cause to enmatter a substance. On the other hand, it also uses the formal cause in order to fashion a material into a specific form. So this is an extremely important part of the mapping that's been left out up to now. So let us look at a few examples. So just to get this formal concept more real. So Aristotle, if you want to translate this into Aristotelian terms, you have efficient cause, mapping the material into the effect using the information in the formal cause. So in fact, if you think of it as a process, the efficient cause is a processor. The input, in order to get input from output, you have to know what the type of process is. What is the information that the processor needs in order to do this particular process? So that information that is needed, I call information. There's also a new term that I want to introduce today. I think it's important. It's the information that the processor uses to inform the output. If you think of an artificial process, and I'm going to use this quite a bit, and I have a 3D printer here in my workshop, so it works very well for me. So what a 3D printer does, it's the processor. The material that it uses is what's called polylactic acid fiber, is one of the types. It needs a description in order to layer all, subsequent layers upon layers upon layers in order to build a particular object. So it's completely analogous to what I've had up here. And then in the cell, you have biological processes, and that's one that we've seen it already. A ribosome makes polypeptides using these amino acid tRNAs, the substrates, but it needs the sequence information of the nucleotides and mRNA in order to be translated into the sequence, the amino acid sequence information in a polypeptide. So you can see this is a very good formal model of all of these processes. Very, very useful way of thinking about these things. However, this type of formulation of the model is a little bit unsatisfactory because you can't figure out exactly how does the formal cause fit in. And in fact, it fits in 4 different ways. And I'm not going to talk about too much. You can have the formal cause being part of the efficient cause, or it can be, it's called an intrinsic formal cause, or it can be an extrinsic formal cause. I'm going to give an example now. Or the formal cause can sit inside the material, in the material cause, or it can be associated with a material cause. Now this sounds a bit funny, but let's have a look. So for instance, an enzyme catalyzes a substrate to a product. The binding specificity tells it what substrate to bind. So it's like this, the lock and key idea. So that is a constraint on what the enzyme does. So we can call this binding specificity is the information, the formal cause. A ribosome is a catalyst that uses a template together, the ribosome plus mRNA, for instance, to specify the monomer sequence inside a polymer. If you think about the functionalizing context, the sequence information inside, for instance, a polypeptide, is the information that is needed for the polymer to fold into the correct 3-dimensional structure. And on the other hand, you can also have something like a tagging enzyme. For instance, you can have DNA acetylation, histone acetylation, DNA methylation, protein ubiquitination. All these are ways of tagging a particular target so that in the end, the formal cause sits in. And you can see the formal cause can enter this mapping in different ways. I'm not going to talk about these now, but I have very nice, we can get to it later, very nice real examples, in everyday examples that match these particular configurations. Okay, Yogi, nearly finished because we're now now you've got.
Yeah. So you are finishing slowly but surely, right?
Excuse my dogs. Apparently you've had them before, so it's nothing new. Okay, so how does all of this fit into what I call the construction, the manufacturing system inside the cell? So here, if you think, von Neumann was, of course, one of the first people to think about this in terms of his universal constructor. He had this idea, and you can see this is exactly the same as this type of configuration. You have a description of some automaton. You have the constructor that uses that description to convert materials into that particular automaton. Or you can, of course, give the automaton its own description, its own information, and then it can construct itself. That's the basic idea of von Neumann's universal constructor. However, real processes usually don't work this way. Real processes usually work by a fabrication where the constructor constructs the parts of an automaton, and then there's an assembler that assembles those parts into a composite automaton. And the idea could, that, of course, be that that assembler actually also can assemble the parts of the automaton itself into the automaton. So this is the idea of von Neumann, but expanded into the idea of manufacture as a process of fabrication followed by assembly, which I think is a much more realistic formulation of what we know in everyday engineering and also what happens inside the cell. Okay, so we start off with that. You can see if I can also translate this into the 3D printer. So a 3D printer can make the parts of a composite object, which is then assembled by an assembler. A ribosome can use mRNA to make a polypeptide, and the polypeptide can be assembled or folded by the intracellular milieu into an active enzyme. So again, these models actually map very well onto what happens inside our own artificial manufacturing systems and in natural manufacturing systems. And I can put all of these together now into a fabrication assembling model that also has this mapping in that creates or maintains the intracellular milieu, as we've said. This translates into this one here. So we have enzymes converting nutrients into building blocks for polypeptides. And then the ribosomes use the information in mRNA to make the polypeptides. The intracellular milieu, through folding and self-assembly, produces active enzymes, ribosomes, transporters, and the transporters then use the extracellular electrolytes to maintain the intracellular milieu. So I'm able then to use this formalism to construct a formal model of what I had before as this three sets of efficient causes working together. This is then using this, I would call a category theoretical model, which captures in a formal way all of those interactions. And of course, I can also then relate parts of these models to the three sets of efficient causes that happen inside the cell. For me, this is not so important. I just want to show this, I'm able to do this. But I think it's the central concepts that come out of this idea of assembly, of fabrication, assembly equaling self-manufacture that are important for our discussion. So Yogi, I'll stop there and I'll stop sharing my screen.
Fantastic, thanks. We give you a bit of leeway because you covered a lot of territory in very little time and very clearly. Thanks a lot for this. And also I'm happy we covered the secret of life, which is to make use of macromolecular biochemistry and weak interactions. Supramolecular, sorry.
In fact,
so
if I may say something there, Yogi, it's not only that, of course, it's autocatalysis because of this hierarchical cycle, which is autocatalytic, and self-assembly. Matches perfectly with Deacon's autogen, which also uses, in order to explain how purposeless systems get to purposeful systems, he has this model of an autogen which uses autocatalysis and self-assembly. So it's a very close match between what he does and what I do.
So there's three things that we want to get across here, and this is one of them. You get purposeful behavior and sort of teleological phenomena from self-manufacture. So this is, you need to understand the self-manufacture first, and then you can understand what Deacon calls these intentional phenomena, and that's very important. The second thing is, I really think this has to be stressed, that it's really useful to have a formal model here to think clearly about the problems and the concepts that we need to understand the specifics of cellular dynamics. And the last thing, that was the most obvious point in your presentation, is we need to have models that are congruent with reality, and which was Rosen's arguably biggest problem. But again, Rosen developed a formalism that was extremely useful, and we have a certain empiricist fundamentalism in biology nowadays that says it's useless to have a develop formalisms like that, for example. And I think it's not. So you've demonstrated that. We are running a little bit late, so I get to talk to Jannie a lot. And so I won't ask him a specific question. I'm really interested in the connection of this model and the dynamics of a living cell. But I will hand over to the other panel members immediately for the sake of time. Do any of you guys have a question?
Then I'll start. So first of all, Jannie, it's cool that you read Rosen, that you read Terence Deacon. I'm happy, and I hope we'll have more follow-up discussions about it. But there are two central questions that I kind of want to ask, and I think they might also be interesting for the audience. And I think the first one is this interesting relationship between category theory and biology, basically, that it is, at least in the relational biology school, but also in some other places now, category theory has been used and is being used currently to model biological processes. And I'm curious if you want to elaborate a bit on that relationship and what makes category theory such a special tool of the kind of modeling tools that we have to think about biological processes. And the second one, the second question where I'd love to hear your thoughts on is this question basically of the emergence of these systems. And since you've mentioned Deacon, I think Deacon spends a lot of time in his work to describe basically how such minimal systems could emerge. And the systems that you've described are quite complex, and it seems very unlikely that these systems just came out of nothing and just showed up with the complexity that you've described, basically. So there were likely minimal precursors to these types of systems. So I'm curious how you're thinking and how the modeling and the tools that you've provided, what insights they give you towards this question, basically, of the emergence of minimal living systems.
Okay, so let's start with the category theory. Of course, this is interesting. So, of course, there's been a whole revolution in mathematics. If you like follow the history of mathematics, most of until about the 1940s and even up to now, one of the foundations of mathematics is the idea of the set. And the most important part of the idea of a set is that you either belong to it as a member or you don't belong to it. Yeah? So set theory is set membership is the basis of the idea of set theory. So in the 1940s, MacLean, what's the other guy's name now? I can't remember now, sorry. But the idea of, in algebraic geometry, the idea was developed that one can actually think of mathematics in terms of mappings between, or morphisms between objects. In fact, you can go further than that. You can only think in terms of morphisms and forget about the fact that the objects are sets. So the idea is that the relationships become the important thing. Yeah? The relationship of one thing to another becomes the important fundamental building block, let's say, of mathematics. And in fact, William Loettgers has tried to create a foundation of mathematics built purely on morphisms or mappings or arrows, or whatever you call them. So an arrow or a morphism is a mapping from a domain to a codomain, or from arrows from a source to a target. But it's always the relationship that is important. And it's, for me, always interesting, why did it come so late in the history of mathematics? Why did it only come in the 1940s? And I think it's our human tendency to try and categorize everything. You know, it's either white or black, or this religion and that religion. You belong to this or this, or this political party or that political party. We tend to put things into sets. It's a natural way of thinking. Our natural way of thinking is not to think of relationships between things. That is something that is added once it's become obvious that one should actually do that. But maybe that's the reason why it happened so late in the history of mathematics. So for that, category theory is very important. You can see in my model, I can't do without these mappings. These mappings are the central part. In fact, it's not so important what maps to map. It's how the mappings are related to each other that actually then creates the model that does something interesting. So maybe, I don't know if that's enough of the category theory, but it's very important. I think this is the formalism that is useful for relational biology. Of course, one uses the category theory. You can have the category of sets and mappings, or you can have the groups and homomorphisms, whatever. There are many types of categories, and the most fundamental one is the category of sets and mappings, and that is why relational biology actually uses that. Okay, so category theory. The second question is this origin of life. Okay, the jackpot question. So of course, biology is very interesting in that there are three questions. The first is, what is life? The second is, how did it originate? And the third is, how did it evolve? And these are three huge, different ways of thinking about life. The definition of life, some people say you can't define life, it's useless to define life, but I disagree. I think it is possible to define life, at least to define life and say, what is the most basic requirement for a living system to be able to do what it does? And my answer is, it has to self-manufacture. And Terence Deacon's answer is basically exactly the same. You have to have such a system before you can change through natural selection. And we can talk a little bit about the idea of open-ended evolution and whether my model actually can explain open-ended evolution. Can it explain the fact that you have to have an open system, thermodynamic open system far from equilibrium in order to be a dissipative structure, Purkhauser's idea. So all of these things are interesting to relate my model to. But what we have to ask ourselves, if we start off with something like Deacon's model, right, and we end up with something that we have now, which is my model, what is it that needs to be explained in between? And I think the most important thing that has to be explained is the idea of, how do you establish a coding system, a coding table between what happens inside protein sequences and what happens inside the information that is needed for making those protein sequences. So the genetic code is another elephant in the room. You can't start only with catalysts. You can't only start with polynucleotides. You have to have a system that combines the two. Now in Deacon's system, the formal causes will not be a separate formal cause. They will have to be embedded inside the materials themselves. But somehow in the evolution of life, those two got split into an extrinsic formal cause, and the activity, the efficient causes. And that, I think, is the crucial question if we want to try and get from there to here, what we have to explain. I don't have it, but I think that is where we should focus to a large degree. Because I think Deacon's model is a very good starting point for the beginning to get to a system that is then able to undergo natural selection, right? Because as he makes the point, you can't have natural selection before you have a self, and so the origin of a self, a purposeful self, is the origin of life question to start off. But from there, you have to then try and see what happened in between until you get to the sort of model that I have. And I don't know, but I think having the two juxtaposed to each other can give us ideas of how to try and, connect somewhere along the line.
Fantastic.
Thanks a lot. I'll pass it on to Kevin or Andrea or Yogi.
Right. I just want to say, Sergio, I see you. We'll open the discussion to the audience in about 10 minutes, okay? But we've seen you.
Andrea, would you like to start? Otherwise, I can go first.
Wait, maybe I have a short question. So I'm more interested in modeling practice. So coming back to Paul's question in the beginning. So when I read your paper, and also when I read Rosen, as far as I could understand it, so one of the questions which came up in my mind is, how much could we, category theory, as you explained, is very important for this kind of modeling. And as you pointed out, it's also a tool in that it forces you to think clearly about something. But on the other hand, how much would it be also a constraint to that, could one not argue, okay, this may be one way of one possible instantiation of this kind of mechanism and self-manufacturing you are seeing? Couldn't there be another mathematics which also, we don't know yet about, but would also fulfill the same kind of thing? So I'm asking this question because I was curious when you were showing Rashevsky and relational biology, and I remember vaguely that he had been also in contact with McCulloch and Pitts, as far as I remember. And they had been, one of them was a logician, the other one, I think, came from psychiatry. So, and they developed something like this network account. So where you also have relations and interactions, and, people moved on, and it came people like Alfred Loettgers, and he developed these differential equations and dissipative systems. And they were much more focusing on the openness of biological systems, and try to capture this part that you are far away from equilibrium. So I don't see, it seems so that these models can do something. Your model is very, very clear and very nice, but it cannot get hold on this part. So it's not a critique. I'm just interested in this kind of, how you construct models and what are the constraints in when we construct. Also, when it comes to concepts, you talk about machine, hardware, software. So how much it also, guides your intuition about how these processes look like.
Excellent question, Andrea. So it's very close to my heart, this. Of course, it depends what question you're asking. So my model answers a very specific question, but there's a hell of a lot about life that it can't do. So my previous life, as Jaeger has said, there I was interested in the kinetics of biochemical networks, right? So there you have, you also have relationships, but you want to know how fast things happen, how they're regulated. What's the effect of feedback, allosteric mechanisms, genetic regulation? There you use dynamical systems theory to answer those questions. I cannot answer those questions using category theory. Category theory, so there's no dynamics in my model. It's just purely a flat representation of the relationships of functional organization. So category theory is perfect for modeling functional organization. It's completely useless for modeling the kinetics of what happens inside that system. Yeah. So Rashevsky had exactly the same problem. He started off, he's the father of mathematical biology. He was doing dynamical systems. Yeah, that was until he realized that formalism couldn't answer the questions that he was asking about organism. That he started actually the idea of relational biology. He was the father of that. Rosen took it much further. But this idea of organismic sets, that was Rashevsky's idea. So he also realized that the formalism that he had been using, the mathematics, could answer lots of interesting questions. For instance, the two-factor system that was in the end then the basis of neural networks, with McCulloch and Pitts. So all of these things, came from there. But you need different mathematics to explore different aspects. Life is very rich, what happens inside cells. So I agree absolutely with you that you need to be sure about what you're asking and then choosing the right tool to answer what you want. And so that's, for me, category theory was the right tool, for Rosen as well. But for my previous life, dynamical systems theory was the correct tool. So you have to, there's a mathematics virtually for everything. As you said, there may be new mathematics that are useful for answering other questions. But I think those are the two. Okay, mathematical biology in general is 99% is dynamical systems theory. very little else. Yeah. And so it's important to realize that there are other ways of also modeling to answer different questions.
Oh, thank you very much for this. This was very helpful. Thank you.
Yeah, thanks also from my side. Excellent and motivating. And I think that your mantra you're using in your papers would fit here nicely with nothing in an organism makes sense, except in the light of the context as it goes, I think, which is brilliant. And also coming to the question of context and maybe context shifts. I think you mentioned it a bit now, but maybe let me start with a shorter technical question when we now discussing if category theory is the right one for this purpose. You mentioned that you started to split F in this one model and then started to split B, but you also thought about splitting G instead of B. And I guess it wouldn't really make a difference after all, but I would still like to ask the question if there are some possible bifurcations or in the adjacent possible of your modeling practice beside the way, the path you just showed us. Would there be another way to go at some points where you could do it differently, still working with category theory and more or less the way you did? And a second question that is dear to my research a bit more and more general and maybe intuitive is I would really love to hear your thoughts about emergence, how you think about emergence itself as a framework or a paradigm or whatever it then is you have in mind. Thank you.
Okay, thanks. So with regard to the splitting of G, of course, I think I discussed this in my paper, and people can come back to the paper to have a look at that. Splitting G is possible instead of B, but again, it leads to infinite regress because, and I don't, it's difficult to explain it without, going to the diagrams, but it has now you have an infinite regress within the hierarchical cycle. Right, so you have to split G, then you have to split F again, you have to split G, you have to split F. So you run into the same problems of infinite regress. The reason why I split B is because that's the model that fits in what I know about happens in the biochemistry. So again, there's always a to and froing. I think this is the wrong idea people get. You have a model, you look for a realization, or you have a real system, and you look for a model. That's not the way it works. It's always a to and fro. Now you have a model, yeah, it doesn't work. Now what happens there? Now maybe I should do this. Maybe So you constantly move between model and system that you want to model in order to fine tune them, so that in the end they become congruent. The idea of Robert Rosen's modeling relation, where you have a natural system with causal entailments on the one hand, and on the other hand, you have a formal system with inferential entailments. And the modeling, what you do when you model is you try and bring those two entailment structures into congruence with each other. Yeah. And it's always an iterative process. So often it is said relational biology starts with a model and looks for a realization, but yeah, that may be one way of putting it, but that's not the way it works. It's always, you have an idea, then you check it, and this, a probing that happens from one to the other all the time, until you find that to your satisfaction, you think you have a good analogy between real system or natural system and formal system. Emergence. I am an emergentist, if you want to. Such a person. I think somebody like Terence Deacon also calls himself an emergentist. Just for interest sake, if people haven't read Terence Deacon's Incomplete Nature, it is a rather thick and dense tome to read, a brilliant book. But there's a wonderful shorter summary of it called Neither Ghost Nor Machine by Jeremy Shermer, which it's a shortened version which captures the idea of incomplete nature very, very well. And he also talks about, emergence. So I think for me, emergence is a beautiful example is this idea of going from a system that has no purpose to a system that is purposeful. Yeah, that is the emergence of function, basically. That's what happens at the origin of life. You go from functionless chemicals, yeah, into a system that now has something completely new that you cannot describe in terms of chemistry. You can only describe it in terms of organization. Right. And so there's a beautiful example of an emergent property. I'm not sure that, so I'm not an expert on emergence. I mean, lots of paper has been wasted on writing about emergence. You have the strong emergentist and the weak emergentist, and then the top-down causation, downward causation. So this is a huge minefield, of which I must say I don't know everything. I'm just a poor biochemist trying to understand what happens inside the cell. So all of this stuff, I've read it, but all I can say is for the intuitive idea of emergence, I can understand it. And for me, that is a beautiful example, the emergence of purposeful systems, which you cannot relate to the nuts and bolts of, you know, in this particular instance, the chemistry of the system from which it arose. So we can have a discussion about emergence as much as we want, and maybe again when we get together in July. But all I can say is yes, emergence is there, and there are beautiful examples of it that I think really fit the concept well.
All right. Okay, thanks. This is, I mean, there has been a lot of ink spilled, but we're going to spill some more ink on the topic of emergence. And I think one thing that's really important here is that this model that you have of the cell is an example of organizational emergence. It is an example of matter being organized in a way that isn't the case in non-living matter, non-living systems. So this is different from what a lot of people think emergence is, like the computational emergence that you get in the game of life, or then the voodoo magic that David Chalmers calls strong emergence. And that's just something he made up in his armchair somewhere, which is really not useful to contribute to the debate. But it's really important to distinguish the emergence of new rules of how matter interacts and computational emergence, which happens entirely within some kind of rule and just means that you cannot predict how the system behaves without actually simulating it step by step. I think this is important to qualify, to clarify at this point here. So this, what you're talking about, is all an example of an emergent organizational state of matter, which is the big question about the origin of life. And I think nobody knows what happened there. Maybe nobody will ever know. So on this, before I start rambling even more, I would like to switch gears and invite everybody in the audience to switch on their cameras, to put questions in the chat, which has already happened quite a bit, and to raise their hands if they have a question to ask on screen. So Sergio has had his hand up for a while, so I would go with you first.
Thank you. Thank you very much, Professor Jannie. So I have three punctual questions. I all the time got confused, or at least I saw in the literature, like people working on the MR system and closure to efficient causation, but as well on operational closure and autopoiesis, or even further in closure of constraints of Mossio et al. That there is a little fine-tuned confusion with feedbacks or feedforwards. So I would like to know to what category of causation, of Aristotelian causation, a feedback belongs. This is the first question. And I have a second question as well. Do you think that a possible biological relational approach exists to the protein folding problem, or could be developed?
Okay.
And well, I will stop there because perhaps the second one is too much. Thank you.
Very good. Thanks for the questions. Nice being able to meet you online. So just interesting. So I didn't mention this before. Of course, there are many people that have thought about this. I mean, this idea of operational closure or closure to efficient causation. I mean, it goes back to Immanuel Kant. Hans Jonas talked about it. And of course, there's a von Neumann, Rosen, Maturana Varela, and Tibor Ganti, Deacon, and then Matteo Mosio, Leonardo Pich, and Alvaro Moreno with a closure of constraints. So these are all important vantage points on the same problem. Constraints, of course, are formal causes. So there's a very close linkage. What I, of course, read all of this stuff, and they get to a point where they stop. So that, you know, closure of constraints. Okay, closure of constraints, very important, I agree. But how does it work? What, you see, that's the question I always ask. How does it work inside the cell? So I acknowledge all of these contributions by all of these people, and very important contributions, but nevertheless, they don't ever go that last step to translate really what happens, you know, what happens inside the cell. Feedback, very important, because that has been, again, a large part of my previous life. So we're talking about feedback in metabolic systems, I suppose, Sergio, are you right? Is that what you...
Yes. In general, but in the whole scientific discourse, like either is immune systems, neural systems, or even at the climate level, we talk about feedbacks. And feedbacks is a central concept of Ashby and all the cybernetician movement. So, what is the...
No, okay, I've got it. So let me use an example of feedback, which you know, for me again is very clear. So you have a metabolic pathway that starts with something, and you have a sequence of reactions, enzyme catalyzed reactions, that produces a product. And that the first enzyme, so-called committing enzyme in this sequence, usually has a binding site for that last product, which either usually inhibits the enzyme or activates it. So there's a feedback loop. So the first important thing to realize, it's not an efficient cause. The feedback is not, the enzyme is the efficient cause, and all that the metabolite does, it constrains the enzyme to do either better or worse, yeah, or more active or less active. So the feedback inhibitor acts as a formal cause. So all feedbacks can be related to formal cause. They are never efficient causes. They act on efficient causes. They act through efficient causes. Yeah. So I think that is very important, because I think people often when they talk about circular causation, they think, you know, that is circular causation, but it's efficient causation, and the circle is via formal causation. It's not via efficient causation. A hierarchical cycle, on the other hand, where you have an efficient cause which produces another efficient cause, which then produces the first efficient cause, like an autocatalytic cycle, that's not feedback. That is autocatalysis. That is closure through efficient causation, not through formal causation. So that's my take on feedback. And then you asked about protein folding. See, the protein folding is, of course, one of the most difficult questions of all, because at the moment, you know that we've just had a Nobel Prize, you know, with AlphaFold, but that's not understanding how proteins fold. That is, you know, that is modeling or mimicking, in a sense, how proteins fold. Simulation. So these are all simulations. So for me, always the interesting point is, if I want to say I understand folding, then I would say, fine, I give you, or you say you understand folding. I give you a long string of amino acids, a polypeptide, and then you have to you have to have a simulation that folds it up into the active conformation, yeah? So at the moment, nobody can do that. So we can predict possible, yeah, but we don't know whether in the first place, whether we have the right structure. In the second place, we don't know what that structure does. So also, you don't know what the function of that structure is. So at the moment, what people do is they do homology modeling. So you look at the sequence of amino acids, and you do the crystal structure, for instance, and all these things interplay, and then you compare it with the crystal structures of other known proteins with known functions, and from there on, you through homology, you try and say that you understand the folding problem, but you don't. So Rosen wrote about the folding problem, was very aware of it, actually tried to figure out a way of doing it. So we're still stuck in the situation that we cannot, so if you want to do the folding, you have to be able to model the intracellular milieu to start off with. Then you have to place in your model, you have to place this long string of amino acids, and then your model has to fold it up into the correct structure. And we can't do that yet. Whether we'll ever be able to do that, I don't know. But I don't think category theory is a useful tool at all for trying to understand protein folding. This is a dynamical process, which depends to a large degree on quantum effects as well that have to be brought in. So, and this whole energy minimization that you have to do when you try and predict the folding, it's a huge minefield, and I leave it to the experts.
Thank you very much. Very useful your answer, Professor. Thank you so much.
Thanks. Great. So I just wanted to stress again how different these models are that you're presenting here. They're a tool for understanding, and how often we mistake models just for their use in prediction. And often now with AI, we have prediction without understanding. Who knows where that's going to take us, but it's definitely not reliable knowledge, and it's definitely not insight, right? And that's what we're after here with these models. So I've seen you, Vasilis. We had a question in the chat that I want to bring to Jannie, and that was from Jordan about whether you could have such a self-manufacturing system based on a basically single but complex molecule, a single molecular system, even simpler than Deacon's autogen. Or to phrase it a bit differently, what is the minimal setup that you need for such closure to efficient causation?
I don't think you can get more minimal than, well, the minimal setups that you have to have some other autocatalytic process. You have to have something that, that causes the closure, yeah? And then you have to have some mechanism that feeds into it. And that's exactly what we have in the cell. So we need autocatalysis, and you also need self-assembly along the line. Otherwise you can't get from non-functional to functional without self-assembly. So I think those aspects need to be in such a minimal model. Whether it's chemically possible to have, even something even simpler than, for instance, the autogen model of Terence Deacon, I'm not sure. It's something that probably in the next phase of my research I'm going to be much more focused on that. Up to now, I've been worrying about what, how do I explain what we have at the moment in terms of life? But of course, at some stage when you understand that, the obvious question is how to go back and see whether one can understand simpler systems that can also have these properties. So I cannot actually answer that question to know whether there's something simpler. It could be, very well be. And so it would be very useful and interesting if somebody developed something like that.
Fantastic. Great. Okay, so Vasilis, please.
Yeah, thank you very much for this talk. It was really fascinating. I had no idea that category theory was useful in biology, although I suspected it very much. Because you see, one of the approaches, the most productive approach to category theory is to look at it as a language. To talk about structure systems in general. They may be mathematical systems, they may be living organisms, they may be physical systems, anything, and study the morphology, functioning, and even dynamics. That's my question. You rule out the use of category theory for dynamics. Well, I realize that you have very, very complex situations in biology, like protein folding, for example. Okay, but I don't think you can rule out category theoretical approaches to dynamics in principle. You mentioned William Loettgers, for example. Okay, many years ago, back in the 60s, Loettgers developed a category theoretic approach to classical mechanics, including dynamics, etc., because there are many kinds and flavors of categories, you see. And a dynamical system can be seen in terms of morphisms in a particular kind of category. There are suitable categories to formulate that. So the question is this: is there any discussion between experts in different disciplines? I mean, people who work on category theory in biology with people who do that in chemistry or in process theory, things like that, because there are many ideas that can cross-fertilize these things. So because I have this, I know this about classical dynamics, and now there's a project going on to generalize that to quantum theory, quantum field theory, general relativity, things like that, in category theoretical terms. And there's a lot of dynamics there, you see. Of course, biology is much more complex. But I mean, we shouldn't preclude this very, very fertile tool in principle. So that's the idea. Is there, I mean, how did you come across category theory, for example?
Okay, let's start with that, Vasilis. Thank you very much for the question. I think it's very important because it is so that there is a category of dynamical systems. I mean, that is certainly part of category theory. The way I came across it is Rosen. Rosen was the first person to use category theory outside pure mathematics. And that was in 1957, 58, 57, 58, 59, when he was working with Rashevsky on his PhD. So his papers, his seminal papers are 1958, 59, and he used category theory. So that's very interesting that he was the first biologist to use it. Since then, of course, there's been a lot of different ways of using it. Ehresmann, for instance, Van Vermeersch and Ehresmann used it in their memory evolutive systems. They used it more for, discussing how various structures can talk to each other and evolve. So I only know the uses of category theory from what I've read in biology. I'm not so sure about the others. I mean, I know it's certainly used a lot now in quantum theory, but not necessarily with dynamics that I know of. So the problem is you can have a category which describes relationships, but if you want dynamics, you have to have a time element added to it, yeah. And so in some way, the category theory can be the basis of the description of a model, but then you have to supplement it with equations that actually describe dynamics that are time dependent. So I'm not too sure about how it is used in various fields, but I'm absolutely sure that you're right in that category theory can be a language, as you say, for also discussing dynamical systems. And it would be very, very, very important that it is actually used in different fields, and I would be very interested to read that. So if you have examples of that, and we can even contact after, then I'm very interested to hear what your ideas are.
I just wanted to throw in Misarovic and Takahara's general systems theory, which is also based on category theory and has a category of general time systems and a narrower category of dynamical systems defined in terms of the mappings through time and the mappings from input to output and so on. That's not specifically biological, but it's kind of useful, I would say, for biology. Yeah.
The snag, Yogi, is if I take my model and I want to add dynamics to it, then I have to now recast it in terms of dynamical, like equations, system of nonlinear equations. And I immediately lose something there because a system of dynamical equations cannot produce its own dynamical equations. And that's what you need. You need to be able to produce a dynamical equation. You also lose the idea of functionalization. You just assume a function. Function is never produced. And so you lose something when you start doing dynamics. can use that category theory for the network structure, but if you add dynamics to it, you lose certain things. You lose the ability to understand certain things.
Yeah, so the insight that we haven't mentioned yet from Rosen is what he called the non-computability of these relational models. And I think that relates, so our argument in the relevance realization paper was that this is not a non-computability. It's not that you cannot compute these models, it's you cannot formalize the future behavior of such a system. And I think you've made similar arguments that are not published yet about collective impredicativity in these systems, and that leading to a fundamental non-formalizability of the future behavior of a system like that. Do you want to say anything about that in terms of also the connection of in introducing your formal cause into the model and getting evolution out of the model, which is not possible with Rosen's original formalism, right?
Sure, I think, so let's take the last one first. I think that's very important to realize that the model that I have is open, and there's open-ended evolution is possible. So if you think about what is let's just call it the primary formal cause in the cell is the DNA, is your genome, yeah? But it is the sort of lookup table that is used by the cell to do whatever the cell wants to do, right? If you want to, I can use the word want because we have a purposeful system, so I'm allowed to do that. And so think about it, DNA is transcribed into mRNA. The mRNA is actually the direct formal cause for making polypeptides. So it's the mRNA sequence that is translated into the amino acid sequence of polypeptides. But the mRNA, before it, from DNA to mRNA, lots of things can happen. You can have alternative splicing. You can have, introns taken out of the mRNA. So lots of stuff happens before the formal cause actually comes to where it is used. So between DNA, primary formal cause, and let's call it secondary formal cause, lots of things can happen. So the cell doesn't actually directly use the sequence of DNA nucleotides. It uses transcription products as the formal cause. But what the cell can do is it can, through variable gene expression, it can adapt within what the DNA allows. So the DNA is sort of a, what is the space of possibilities for function in the cell, but the cell can use that dynamically. And we know we have different cell types that express different parts of the DNA. The DNA can, of course, change. So my formal cause, which is at the top, can change through mutations. It can change, for instance, by gene duplication. It can change by, for instance, some bacteria, under high stress conditions, they switch off their DNA repair mechanism so that suddenly they start producing a whole, set of variants in order to, perhaps the idea of one that possibly work under these stress conditions. So there are all types of ways in which the DNA can change or can be used in changeable ways. So you can, the model allows adaptation within genomic constraints. It also allows adaptation over lineages, which is natural selection. So in that sense, it's open to formal causation. Yeah. However, what's interesting, the model, as I described, is blind. It's blind to its external environment. But that's no problem, because the way that cells are, if you want to talk about primitive cognition, the way that they sense the outside environment is through receptors on the membrane. The receptors are proteins that are made by the self-manufacturing process. So there's no problem with extending the model to include sensors towards the outside of the environment.
Yeah, I was just going to say, you have nutrients, you have electrolytes in your model, so it's just at the single cell level, right? And that's another, how do you extend this to multicellular organisms? Is a multicellular organism like a tightly integrated ecosystem? What distinguishes it from just an ecosystem or a community of bacteria and so on and so forth? And how do we deal with this idea that closure actually comes in degrees in these situations when you go above the level of the cell?
It's that's a topic that I, don't have a direct answer. I mean, obviously, you as an organism are also close to efficient causation. Yeah, you produce yourself as you sit there. You eat food, the food becomes you. Food and the waste products go out. So you have a constant flow of matter through your system, but the self remains. Your selfhood is intact. Your mother still recognizes you after a year, although all the mass has been translocated from outside through you to the outside again. So in a sense, these concepts have to translate up to the full organism as well. But then it becomes a question of looking at not what happens inside the cell, but what happens on an organ level, for instance. Yeah. So if you're a developmental biologist, you know, as cells start differentiating, they have different functions, and they, and you can find a higher level of description, probably also with category theory, that shows how these things are related, how the different organs, are related to each other, what they do, what their functions are. So this, as Vasily said, it's a language that we can use for discussing relationships at whatever level. I've now concentrated on the biochemical level in the cell. We can use this idea of what happens inside the cell. You can translate it up to very interesting higher order structures like businesses or societies or economies or whatever. Yeah? So again, in all of these things, functional agents that need an enabling environment in order to function. Yeah, so you have to train them, you have to metabolize them, you have to train them into a particular state. Then they must become functional. If you have a toxic environment in a society, then this culture doesn't work. Yeah, so if there's something wrong with the culture, people can't function, they can't fulfill their roles. So for me, what the cell, this organization has done is allowed me to think in terms of these Aristotelian causes, what happens in other systems as well. And of course, in a complex organism like us, the same language can be used to describe the functional relationships, the functional organization in the system.
Yeah, so the problem right now is the lack of coherence at a societal level, not a closure that we have. I don't like these superorganism metaphors for society at the moment because of that. But if there are no more questions, yeah. Sorry, go ahead.
No, I don't think of them as superorganisms, but I think that the concepts and the language is is interesting.
Yeah,
because some of them nicely up to other levels.
I wanted to ask if there are further questions from the audience. We have time for one more. If not, I have one, and that is so we talked about the relational model and dynamics. How about relational models and thermodynamics? How are these models related? How are they compatible with what we know about far-from equilibrium thermodynamics theories like Prigogine's and all these kind of things?
But that's extremely important because, again, the model doesn't have time in it. It also doesn't have energy in it. But if you look at what happens in the description of the model, it's perfectly compatible with open systems. It is open. So the nutrients on the outside, the electrolytes sit on the outside, yeah. They are used, so there is a flow of material through the system. Of course, my model at the moment doesn't include the breakdown of things and waste products and whatever, but I can easily put that in. That's no big problem. Doesn't do anything to the model, just enhances it. But so the model is possible. It's possible to think of the model as a conduit for energy flow through the system, which is needed to take it from, or to keep it at far from equilibrium conditions. So it is thermodynamically open. That's important. It's open to thermodynamically open. It's also open to formal cause, which is the other important thing. Yeah. So open-ended evolution is possible, and maintenance far from equilibrium is also possible. The model doesn't preclude that at all. It's just that it doesn't at the moment make it obvious how the energy equivalents come in. So one thing that living systems do is they can buffer themselves against lean times. So for instance, bacteria take in glucose and then they build up glycogen reserves. So that if there's no more glucose inside, they can use internal glycogen as a source of glucose. So they suddenly they are still open, but they're not open to the outside. They're open from the inside for flow. Of course, when that gets depleted, then there's no more food and the organism can die. But all sorts of interesting things the cell can do to buffer itself against coming to equilibrium, which is death. Yeah.
So the tricky thing about dynamical models of whole cells is that you basically have to have a system that rewrites its own equations, because that is a crucial ingredient. It's not just optional. It is an ingredient of what this self-maintaining, self-manufacturing organization is about. And these Rosen's diagram, but also more recent diagrams of just network diagrams are sometimes a bit misleading, because it is really crucial to have not just dynamics underneath of the processes that interact, but a true dynamical organization itself that is constantly emerging. We're back to the topic of emergence. Okay. So before we get into that, I would like to thank everybody for attending and contributing, and Jannie for doing a masterful job in just a bit too much time, but absolutely huge amount of territory you covered there. And we have Evan Thompson as our next guest next month. So tune in again if you can. And thanks everybody for joining here.
Thanks, Yogi.